in their phenotypic presentations. And I am told that I'm the only one that can tell these guys apart, and you may have noticed that not all of the species were described in the distribution slide that I put up. But all of these species are in some way morphologically diagnosable as distinct taxa. And while you may not recognise the different species as different, we get the presentation of two gross morphotypes that look like they're adaptive in an ecological sense in two manners. And the first relates to species body shape. This little critter up here, you can see he's got a shorter, fatter body type, he's got shorter limbs and a shorter tail. We seem to get this presentation of body shape on sand plain habitats. The bottom little lizard here, he's longer tailed, he's got a longer limb, and he's generally really skinny and spindly. We see this occurring repeatedly in species that occupy sand dune habitats. Another aspect of adaptive diversification, or suggestion of adaptive diversification, we see are the colours and patterns on the dorsal surface of each of these species. Now, this lizard here is kind of brown and yellowy. We see brown and grey dorsal patterns and dull markings on species that occupy lighter sand colours, whereas we see less ornate but brightly coloured species that occur on the redder, oranger sands. And so this kind of thing suggests to me that this group might be a good way to test some hypotheses about adaptive evolution across the arid zone. And if this is in fact an adaptive divergence, there is a number of expectations that we would see in this group. And the first being that speciation has been driven by ecological opportunity. Namely, we would expect to see key diversification and speciation events that could be associated with the development of deserts in Australia, which occurred roughly 2 to 4 million years ago. We'd also expect to see perhaps diversification in species associated with the key period here, dune development. And this period is not only associated with dune development, but the diversification of different habitats across the arid zone. Another expectation we might see is that key ecological innovations have occurred at different points in the species history. Namely, that you might observe divergence in particular niche characteristics at different hierarchical levels. The third expectation would be that ecological opportunity has driven phenotypic divergence. Namely, you would expect to see a correlation between ecological and phenotypic divergence in the species. So now to the first expectation, that speciation has been driven by ecological opportunity. What I did was I gathered a series of multi-locus sequence data, again representing both mitochondrial and nuclear sequence loci. I chose samples to represent both the genetic diversity and the geographic diversity within each species and across all species. I then I wanted to get some estimated of the timing of diversification in this group. And so a first step that I wanted to take is get some sort of estimate of rates of evolution in a mitochondrial locus. And I'll explain why I did that in a minute. But how I did it was I took a series of sequences from GenBank that represented the entire Agaminae subfamily. I used a series of fossil calibrations to calibrate that species and estimate divergence, and correlated that with other published records for the Agaminae subfamily phylogeny. I then calculated a median rate of evolution for the mitochondrial locus that I used. I use that locus because it's the most widely sampled across that distribution of taxa, and I could use the fossils to calibrate it. I then took that median rate and I plugged this into to time calibrate a species tree analysis. Now, the reason why I wanted to use a species tree analysis rather than a standard phylogenetic approach was that I had some evidence that the differences, there were incredible differences in the topology suggested by the different gene regions that I'd seen. And this is a very common process in very rapid and recent radiations. What you get is large amounts of incomplete lineage sorting, and this can be exemplified here by the blue gene. This shows that these two species have completely sorted, whereas the topology for this red gene is completely different. And species tree methods seek to sort of average across that to obtain the actual relationships amongst species. And so using this approach, I was able to get an idea of both the timing of diversification and the relationships amongst the different species. This is the species tree I got from a program called StarBeast. Now, the exact relationships amongst the species are not particularly important just yet. What I want you to note is that we have a time axis along here from the present through to about 3 million years ago. We have posterior probability support values on each of our nodes. A value of 1 represents 100% support for that particular relationship amongst the species. What I also have here are these grey bars. These represent 95% confidence limits around my estimates of divergence amongst the different lineages. And when we look at our particular events, we were interested, firstly, the derivation of Australian deserts. We have the derivation here starting at 2 to 4 million years, and we can see that the 95% confidence limits for the derivation of our entire group are entirely nested within that period. Additionally, when we plot the formation of dunes in this system, we note that every single of the recent tips and diversification in the species group have 95% confidence limits which overlap with this period. And what this suggests to me is that speciation has in fact been driven by ecological opportunity in the system. We've been able to link key events in the development of the arid zone with both the derivation of the group and speciation in recent lineages. So on to the next expectation, that we've got key innovation, ecological innovation at different periods in the group's history. And to do this, what I did was I used the distribution, the spatial positions of each of my samples. I extracted a series of climate, soil, vegetation and elevation data from publicly available sources. I then used a principal components-based technique to quantify niche divergence amongst species pairs. And the particular method I use is being described in this paper here. I'm not going to go into the details of that method. I'm happy to discuss it with you after if you'd like to talk about it. One thing that is important though is this particular method uses the loadings of a particular axis, a principal component, to allow a biological interpretation as to what that particular axis is telling you about the niche of different species. And I'm going to talk more about that in a second. I then compared the different patterns of niche divergence at different hierarchical levels across the species tree in order to understand what factors might be associated with both the invasion of the arid zone and then divergence amongst species within the arid zone. And to do this, I used the species tree that I showed you before. And when I looked at this, what I found was that when I compared species that predominantly occur in the wetter, mesic, semi-arid regions of the southwest of Australia to the rest of the group that occupy more arid, northern, central and eastern areas, was that there was an overwhelming pattern of divergence in the thermal niche of species. When I looked deeper into the phylogeny, what I observed was that there was divergence in soil, vegetation and precipitation niche axes, and not thermal niche. And what this suggested to me was that we did in fact have some key ecological innovations at different points in the species history. So lastly, what I wanted to look at is whether or not ecological opportunity could be inferred as a driver of phenotypic divergence in these organisms. And I did this by testing for a correlation between ecology and species phenotype. Firstly, I calculated a distance matrix of ecology based on the principal component scores that I used for the divergence. I then had a series of phenotypic data based on morphometric analysis. And I split these into two data sets. The first describing differences among species in their shape, and the second dealt with differences in species detailing colour and pattern and markings on because I wanted to then use a partial Mantell test method, and I used the distances to account for a shared ancestry between the different species and account for relationships in phylogeny in a partial Mantell test to test for the correlation or control for phylogeny and testing the correlation between ecology and species phenotypes. What I was able to see firstly I had to analyse the data of males and females separately because of dimorphism, which is common in agammons. What I found was that the dorsal markings, there was a significant correlation between species ecology and their dorsal markings when controlling for phylogeny in both males and females. For body shape, the pattern was a little bit more obscured in that species body shape in males was significantly correlated with species ecology, but not for females. And this could be a sampling artifact, it could be a limitation of Mantell tests, and I'm actually going to be using some regression general linear models to look a bit further at this and analyse this a bit. But nonetheless, I think that there is actually now evidence that we have ecological opportunity as a potential driver of phenotypic divergence. And so this represents one of the first quantitative analyses of not only when, but posits how species might have been able to move into and diversify across the Australian arid zone. Mainly, I've been able to show that the derivation of the species complex occurs entirely within the period of aridification and the formation of deserts in Australia. I've also been able to show that perhaps divergence in thermal niche has been important for species moving into the arid zone. I've further been able to show that recent speciation in this group has been associated with initial dune development in the Australian arid zone, and that perhaps divergence in precipitation, soil and vegetation niches may have been important for diversification within the Australian arid zone. In a more general sense, what I think this shows is that adaptations allowing invasion of and diversification within arid zones may be very many and complex and may involve many different processes at different hierarchical levels. I'm now going to change tack a little and talk a little bit more about some of the projects that I'm starting to collect data for in my current postdoc at Yale on Galapagos tortoises. Now these are in a similar vein, looking at the interaction between phenotype ecology and evolutionary history. So within Galapagos tortoises, we have the evolution of two gross morphotypes. The first are the dome tortoises. You can see them across the archipelago here in black. They're characterised by this very round shape. They tend to be larger, blacker, and according to Darwin, have a better taste when cooked. These species tend to occur in the upland areas of the archipelago in the more mesic habitats. This handsome fellow, Wasland Lonesome George, he is representative of our saddleback tortoises. These guys are often a lot smaller. They also get extinct quicker. We can see they occur here across the archipelago. They occur on the flatter, more arid island habitats. And one of the hypotheses sort of floated for the evolution of these different carapace shapes is that the saddlebacks have evolved in arid habitats, this sort of extension of the front of the carapace to allow greater trajectory for the species to browse higher up on different plants, because in more arid habitats they need to acquire more food, there's less plant material around. Another hypothesis that's been sort of suggested for this is that in these habitats, low resource habitats, you get a lot more intraspecific competition. And in fact, the behavioural evidence seems to suggest that saddlebacks are in fact more aggressive and do have a lot more intraspecific interactions with one another. We know that the tortoise carapace shape appears, at least initially, based on our current understanding of the relationships among species, to evolve multiple times on different islands. However, our phylogeny is currently based on mitochondrial data only, and mitochondrial data can suffer we do have some evidence from looking at subfossil remains that haplotypes that were previously present on islands have completely been washed out by a haplotype that was related to a different island than the original one. And so I'm kind of worried about looking at this simply based on this marker alone. And so what I've been doing is working on creating a species tree for this particular group. I've used some genomic data and other population level genomic sampling and utilising the Lonesome George genome that we have a preliminary assembly for to design a series of variable nuclear markers to look at this. And we currently have 27 that we're starting to collect data for. I'm going to use this with a series of environmental data that I've gathered, some high-resolution satellite imagery as well as some climatic data, along with some morphological data that I have with a collaborator of mine, Eleni Chowri. She's developed a photographic method for photographing around a tortoise and reconstructing a 3D model of tortoise carapace shape, which we can then use geometric morphometrics to extract information about the shape of species carapace shapes. And I'm going to be using these 3 different data sources to test the hypothesis that carapace shape in Galapagos tortoises is adaptive in nature. So while we have these gross differences in tortoise carapace morphotypes, there's also much finer scale variation in the shapes of tortoise carapaces that we see. And Southern Isabella is a perfect example of this. We have 2 currently described species on Southern Isabella. The first is Chelonoidis gunteri. It's described from this eastern portion of the island. We have Chelonoidis vicina that's been described from this western portion. These are the 2 different volcanoes, Sierra Negra and Cerro Azul. And Fritz again hypothesized that the development of these different carapace shapes was adaptive in nature. And so what I wanted to do is I wanted to take advantage of a series of blood samples that we have in the collection at Yale to look at perhaps whether or not we could identify the species boundaries between these two carapace shapes. So I took these samples and generated a data set from some microsatellite loci, and lo and behold, we do not have 2 groups. So what we have here is a structure plot here. Each of these bars represents an individual. The different colours represent different genetic populations. The grey is exclusively this eastern population. The white here is exclusively, or the pure white here is exclusively this western population. And these in here represent this entirely novel black population. So we were like, what's going on here? And when you go back to look at some of the literature, often these are anecdotal notes from explorers that visited in the 1900s. What we see is that we actually have a 3rd morphotype here. This is called the Aplastados haplotype. It's described as a tabletop-like carapace shape. We know it's here, and while it was previously said it's actually here, it's actually only in the upland areas. And we think it might be that the really big tortoises get this shape, and it might be allometric in nature. We did think that it might be here. Well, there's some evidence that it used to be here, but it was wiped out by oil hunters in the early 1900s. In addition to this, this sort of 3rd morphotype, there's some anecdotal evidence that you actually have the presentation with the 2 other morphotypes, sometimes within a site. And so there's this incredible variation that is potentially across the archipelago. Unfortunately, the samples I utilised had no spatial information, and they had no information on the shape of the individuals that they were collected from. And that's important because we know that this environment is also incredibly variable. We have a very strong gradient precipitation and vegetation seasonality that runs from the east to the west and from inland to upland areas. And so what I've been doing is I've just started collections here and here. We're collecting data for neutral markers for SNPs. We're also collecting some RNA samples to perhaps one day, as a pipe dream in the future, do some transcriptomics. We're collecting spatial information, and we're using the photographic technique of my collaborator to reconstruct 3D models of the shape of the individuals we collect data for. So hopefully we'll be able to have a better understanding of what's going on here once we collect those data. And so what I hope I've been able to show you today is the power of an integrative approach to understanding both the biogeographic history of organisms and communities and how that relates to their environment, in addition to understanding this interaction between ecology, phenotype, and evolutionary history. And by studying this interaction between ecology and evolutionary history, I hope I've been able to show you that species-specific responses to environmental change is a potential driver of diversification amongst populations within a species. Additionally, by looking at this interaction between ecology, phenotype, and evolutionary history, I've been able to show that adaptations to aridity and then diversification within arid systems may be many, multifaceted, and extremely complex. So this brings me to talk to you a little bit more about my ideas that I have for future research questions. And I want to start talking about this by talking to you about how awesome Texas is for somebody that's interested in looking at the effects of taking an integrative approach to understanding these interactions between ecology, evolutionary history, and species phenotypes. And that's because you sit on an incredible environmental gradient here, and this is exemplified by the average annual precipitation bands that we see across Texas. Consequently, at least in the groups that I'm interested in, you see a composite of both eastern and western fauna that represent very different climatic optima, and I think this offers an incredible opportunity to look at adaptation across an environmental gradient. I'm extremely interested in developing local systems to look at this, and one group I've been looking at in particular are the Sceloporus lizards and allied taxa. We already have some evidence that these groups are diversifying in response to different ecological environments and opportunities, but again, we lack the quantitative tests to show exactly how and why. I'm also interested in getting involved in local conservation projects, and one of the critters that I've been thinking a lot about is the Texas swan lizard. It's been declining for different reasons or different hypothesized reasons, but to my knowledge, nobody's really sort of looking at this in an integrative and quantitative manner, and that's something that I'd like to look at. In another sense, Texas offers an enormous opportunity to continue to study the evolution of and adaptation of organisms in arid environments, because you sit right on the doorstep of the North American deserts. And some of the questions that I'm interested in asking in these systems are, how do taxa representing different lineages, evolutionary histories, ecologies, and morphologies vary in their response to arid system development? And asking this question takes an inherently comparative approach by looking at different species within particular systems. Another question that I'm interested in is looking at what key ecological and phenotypic innovations may have facilitated diversification in arid regions. And I showed you some of the stuff I've done in the Australian system. I'd actually like to take a bigger picture comparative approach and look at how this plays out in different arid systems across the world. I'm also interested in what role temporal and spatial dynamism has played in shaping the evolution of taxa in particular arid systems. And most particularly, I'm interested in semi-arid systems for this, and looking at taxa that occur on the edges of intensely arid systems, but that may have experienced this intense fluctuation in both spatial habitat distribution and climatic extremes. And I think these three questions link into some bigger questions in evolutionary biology. Firstly, how have organisms adapted to arid environments? And I think we can use that understanding of how species may have adapted in the past to link into what might be the potential impacts of climate change on arid biota and other biota newly experiencing arid habitats in general. And so what I've done is talk to you about my research in two different areas, which I'd like to expand taxon sampling in. The first being the Australian Arid Zone. I told you about this group here. I'd like to continue working in other different sympatric organisms. I'm currently working on the Galapagos taxa, the tortoise. There are data sets that exist from marine iguana and lava lizard, which represent different life histories and ecologies. Two of the most unknown groups in the archipelago currently are the colubrid snakes and the geckos, and that's something that I'm actually starting to make headway on starting collection of samples in the system. I've also recently developed a collaboration with Mariana Moriendo in Patagonia. I'm working on the Lyell limus lizards in that region. Lyell limus lizards represent one of the most, in fact, the most ecologically diverse terrestrial radiations on the planet. And they offer an enormous opportunity to study adaptive evolution to different environmental conditions. And of course, the other system I want to start working in is in the North American deserts, and I think Texas would be an exciting place to do that. And on that note, I'd like to thank my late major collaborators. Thank you for your time, and take any questions.